Okay, so this is the continuation of the ABO blood group. So we're done with that. Okay, now we have your RH blood group system. So your ABO, we all know that it was being discovered by Landsteiner at locus 9, chromosome 9 of the locus 9. Okay, chromosome 9. Now, it is also Landsteiner who were able to discover the RH blood group on chromosome 1. Okay? So this one is found exclusively on surface of RBC, the integral part of the red cell membrane, and this the primary antigen, if present, considered Rh positive. Okay. Now there are lack corresponding naturally occurring antibodies in the serum. So we have several nomenclature that was being used to identify your Rh blood group. First and foremost, we have the Wiener nomenclature. So, it has a multiple allele hypothesis. So, in Wiener, sinabi niya there are 5 antigens that are available. You have your RH subscript O, RH small letter ha. So, yung RH subscript O, capital R, H, and subscript O. Okay? You have your small letter RH superscript 1, RH prime. Okay, followed by RH2 prime, you have your HR prime, and you have your HR2 prime. Okay, take note that this is a single locus inheritance system with 8 alternate common alleles coding for agglutinogen. Okay, and one individual may produce 2 agglutinogens that are inherited from both parents. Okay. So another classification is your Fisher race. This is the more common used classification, yung Fisher and race or Fisher race. Okay? So yung pong si Wiener 5. Okay? Si Fisher race 3 alleles. Okay? It has 3 alleles. DD, that's capital D slash small d, CC, capital C and small c, and EE, capital E and small e. Okay, so tatlo lang, DCE, both in capital and in small letters. Okay, also, he said that there are 5 antigens that are available. You have your capital D, C, and E, and small letters C and E. When there is the presence of small d, there is no D-glucose. Therefore, there is no antigenic product. Now, ngayon po, all the Fisher race is commonly used, but what is more accepted is the Rosenfeld. Okay? This is numerical system. So, kay Rosenfeld, ang ginamit naman niya ay RH1 to RH5. Okay? So, Rh antigen, there are 3 integral membrane proteins. These are the RHD, okay, RHCCEE, and Rh-associated glycoprotein. That's Rh50 and RAG, the RHAG. The D antigen resides in the RHD protein. Okay, this is most immunogenic. Okay, so the D is the most immunogenic. Therefore, it can stimulate immunologic reaction. Okay, which is followed by small c, then capital E, capital C, and then small e. Okay? So, yun yung pagkakasunod ng immunogenicity ng inyong antigen or ng D antigen. You have your weak D antigen or the DU variant, that is your RHO variant. This could be weak or absent red cell agglutination by anti-D detected only with the use of anti-human globulin reagent, the AHG reagent. Okay? This one uses the bovine anti-D. Okay? We can form by one of 3 situations. So, sabi natin, Dean, bakit merong weak D antigen? So, it could be there is a missing piece of the D antigen, kaya siya naging weak. Or the D gene is on the chromosome opposite of a C gene. No? So, there is positive steric hindrance. And inheritance of gene coding for less D antigen. Okay? Now, in the blood group system, when there is presence of D, therefore, there could be a presence of the RHO factor. Therefore, the patient or the person is classified as RH positive. But when there is an absence of D, yung capital D ha, yung immunogenic, you classify the patient as RH negative. Okay? Now, how do we test for RHOD antigen? Simply by using an anti-sera which is originated or which comes from a human source. Okay? Anti-sera with different constituents. It uses a high protein media which is necessary to produce agglutination since antigens are an integral part of the red cell membrane. Now, this is less numerous than the ABO blood group. Now, how do we do testing for the DU variant? We use your bovine or albumin suspended anti-D reagent. This is the one that is more commonly used. Sabi ko nga, this is IgG, so it has to react or the reaction should take place at 37 degrees C. Okay? And before you release the result of the RH, it should be done, the procedure should be done in 50 to 60 minutes. Okay? That is to give time for the antigen-antibody complex or agglutination to form. Okay? So, if there is the DU variant, yung DU variant, yung weak variant. Okay? So, pag weak variant, you have to incubate it at 37 degrees C for 15 to 16 minutes. And then, if it is positive with that, then you consider RH positive. There are some women who may appear to be RH negative and should be proven that they are DU variant negative as well. Okay? Before you can consider them to be eligible to receive transfusion. Especially kapag buntis na sila. Kailangan ma-prove mo talaga na negative sila. Okay? So, RH antibody are not naturally occurring. Okay? Immune bodies, this is produced upon sensitization. The IgG isotype, reactive at 37 degrees C, as I have mentioned. The reaction may be enhanced with enzyme treatment. Okay? This type of IgG can cross the placenta, of course. And basically, this is associated with hemolytic transfusion reaction of the newborn. Okay? So, what could be the reason why there are false positive results in RH typing? First, you have the drying. So, kailangan, pag nag-drop ka kasi ng blood, kailangan gawin mo agad kesa ma-dry yung blood doon sa slide. Okay? Number 2, if there are RLU formation. Okay? 3, if there are autoagglutination, and if patient's red cell heavily coated with antibodies, and the presence of cold agglutinin. Okay? Now, why do we have a false negative result? O hindi naman pala siya RH negative, pero pala positive siya. Because you have used old cells. So, primarily, what is recommended when you do your RH typing, kailangan freshly extracted yung blood. Okay? Wrong cell concentration. Okay? Hemolysis, inadequate mixing of cells, inactive typing sera, incorrect temperature, as I have mentioned, again, 37 degrees C. Or, it could be the existence of the DU variant or the weak group, no? And high concentration of blocking antibodies. So, done with your RH, we also have your minor blood group system. Yung minor, hindi to common, ha? Hindi to routinely done in the laboratory. Determination of this minor blood group is not commonly done in the laboratory. So, when do you do your minor blood group system detection? Of course, if there is a medical legal parenthood studies, kung meron tayong parental dispute, okay, we do your minor blood group system. And if, for example, tama naman yung blood type na sinalin mo, pero nagkaroon pa rin ng transfusion reaction, then it is noteworthy that we do the minor blood group system or minor blood group detection. Okay? So, we can classify this minor blood group into cold-reacting antibodies. So, pagka cold-reacting antibodies, these are antibodies form, react at temperatures of 25 degrees C or colder. So, ito yung mga IgM. Kalahi ito ni ABO. Okay? These are not considered clinically significant since any reaction seen in tissue will not be seen in the warmer temperature of the body. This is not likely to cause a transfusion-related accident. Okay? So, ito less likely siya na mag-interfere sa transfusion mo during transfusion. So, ano ba yung mga cold-reacting antibodies? The LE system or the Lewis system. Ang antigens of the Lewis system is LE superscript A and LE superscript B. Okay? So, this is formed in the secretions and absorbed on the surface of the RBC. Antibodies often encountered in individuals with no antigen. This may be present at certain time. Example, pregnancy, and then usually it disappears. Transient lang siya. MNS system. The MNS system is the antigens that are weakly antigenic. So, it is less likely to produce an antigenic response. Okay? So, antibodies naturally occurring or stimulated by direct exposure. Another one is the PP system or P1 antigen. Most antigen. In the PP system, the most antigenic antigen is the P1. Okay? This is present on 75% of the whites and 94% of the African Americans. The II system, the antigens capital I and small I, both present in all individuals. The capital I, present in large quantities in adults, while the small I antigen, present in large quantities on cells taken from the umbilical cord. Okay? And the anti-I, the capital I, this is frequently seen in serum of patients with recent infectious mononucleosis. Again, infectious mononucleosis. Okay? Anti-I, frequently seen in serum of patients with recent infectious mononucleosis. Now, let's go to system that reacts with warm situation or warm reacting antibodies. Reactive at 37 degrees C, this is basically clinically significant. Bakit? Kasi ang ating temperature within, it runs between 36 to 37. Yung normal na, yung sa loob. Okay? Now, reactive in 37 degrees C in anti-human globulin medium. Okay? Clinically significant kasi madali siyang magre-react dahil nga 37 ang temperature ng body mo. This is most likely to produce hemolytic disease of the newborn and hemolytic transfusion reaction. So, kung meron ito, malamang magkaroon ka ng incompatibility. So, first on the list is the Celano system, the Celano system, the capital K and the small k for Celano. Okay? The small k antigen present in 98% of the white population. Antibodies primarily is IgG. The KID system, the antigen present is the JK superscript A and the JK superscript B. This is not very antigenic. However, it can somehow cause minimal hemolytic transfusion reaction. Antibodies is stimulated by direct exposure via pregnancy or transfusion. You have your Duffy antigen or Duffy system. The antigens present is the FY superscript A and the FY superscript B. Okay? Antibodies is stimulated through direct exposure also. This can cause HDN, that's hemolytic disease of the newborn, and HTR, hemolytic transfusion reaction. The funny thing about Duffy system is more common to sa mga negro. And pagka may Duffy system ka, may Duffy blood group ka, nakakatuwa kasi you are resistant to malaria, specifically to P-Vivax. Okay? So kapag ka merong lamok na may dalang P-Vivax, bahala ka. Kagatin mo ako kung gusto mo akong kagatin. May Duffy ako. So, I am resistant to it. Okay? The hemolytic disease of the newborn, that will be discussed on a later date, okay? Not now. At least you have a glimpse of the other blood groups. So, yung mga minention ko, yun yung most common. But, there are also a lot. May Sienna, may Diego, Marami pa yan, but that will be discussed in detail on a later date. But for now, those are the most important other blood groups that which may or more commonly causing HDN and HDR. So for now, have a nice day. Thank you very much, and see you soon. Okay, Happy New Year.